This is a chapter describing the biology and function of stem cells within the central nervous system. This chapter is authored by Phil Horner, Sean Barber, and Samuel Emerson. While it was previously thought that neurons formed only during development, research beginning with papers published by Joseph Altman in the 1960s have demonstrated that neurogenesis in fact continues throughout adulthood in the mammalian brain. These new neurons rise from neural stem cells, and like all stem cells, neural stem cells are defined by their capacity to self-replicate and also to differentiate into specialized cell types. In the case of neural stem cells, differentiation can lead to the formation of neurons, oligodendrocytes, or astrocytes. Neurogenesis has been documented in several neurogenic niches within the adult mammalian brain to varying degrees, but the two most studied are the subventricular zone, which is adjacent to the lateral ventricles, and also in a portion of the hippocampus known as the subgranular zone. The subventricular zone is a layer of proliferative astrocytes found along the lateral aspect of the lateral ventricles. These slowly dividing astrocytes are separated from the ventricular lumen by a layer of ependymal cells, and they extend apical processes through this ependymal layer that directly contact the ventricle. The astrocytes within the subventricular zone differentiate into immature neurons, which are called neuroblasts, and these then travel to the olfactory bulb via the rostral migratory stream, where they differentiate into mature neurons. But the astrocytes within the subventricular zone are also capable of generating more astrocytes, or oligodendrocyte precursor cells, and even myelinating oligodendrocytes. These astrocytes within the subventricular zone are sometimes called B cells, which are colored blue on the graphic below. When dividing, they do so asymmetrically, creating another B cell, just like the parent, as well as a smaller C cell, which is seen in green here. These C cells then give rise to migrating neuroblasts, or A cells, which are seen here in red. These neuroblasts then migrate to the olfactory bulb to become neurons, as mentioned before. The other primary neurogenic niche in the adult brain is the subgranular zone. This area is in the hippocampus between the dentate gyrus and the hippocampal hilus. As is the case with the subventricular zone, the primary neurogenic precursors in the subgranular zone are astrocytes, and they are also referred to as B cells. In the subgranular zone, however, these B cells give rise to D cells. These D cells generate excitatory granule cell neurons that travel to the granule cell layer of the hippocampus and integrate into the circuitry there, extending axonal projections known as mossy fibers into the hippocampal region CA3. Neurogenesis is also seen in other areas of the central nervous system, particularly in the context of injury. Striatal astrocytes have been shown to give rise to neuroblasts in the rodent brain in response to stroke, for instance. Cerebral cortical astrocytes have been shown to generate multipotent neurospheres in response to stab injury. And ependymal cells within the rodent hypothalamus can be seen to exhibit some neurogenic potential in response to a high-fat diet. Neural stem cells are also found near the central canal of the spinal cord. These particular neural stem cells appear to be gliorestricted in vivo, although they are capable of forming neurons in vitro or when transplanted into the neurogenic dentate gyrus, which is an example of the degree to which extrinsic factors such as microenvironment can influence cell fate. Whereas adult neurogenesis seems to be restricted to certain neurogenic niches within the brain, gliogenic precursors appear to be relatively widespread throughout the brain and spinal cord. These glial progenitors are capable of differentiating into astrocytes or oligodendrocytes and also form multipotent neurospheres in vitro. They lack the self-renewal characteristics of neural stem cells, however, and thus have a limited capacity to divide. Multiple distinct types of glial progenitors exist, and each is identified by their potentiality and/or the expression of certain surface markers. The most common of these are cells positive for something called chondroitin sulfate proteoglycan neuron glial 2, or so-called NG2 cells. There are also cells positive for platelet-derived growth factor receptor alpha and cells bearing the surface ganglioside A2B5. NG2 is expressed by oligodendrocyte precursor cells, but is also present in a distinct form of differentiated glia. And cells isolated by NG2 can be induced to form not only oligodendrocytes, but also astrocytes and neurons in vivo. For this reason, they're sometimes called polydendrocytes. They have a potential role in maintaining myelination during aging, but their role is otherwise unclear. Like NG2, PDGFR is often used to identify cells with oligodendrocyte precursor cell features. Mice who are null for PDGFA, for instance, demonstrate reduction or even elimination of oligodendrocytes altogether. A2B5 is a cell surface ganglioside sometimes used to identify glial progenitors, but it's also found on neuronal progenitors and certain types of astrocytes. A2B5 is often used to identify a bipotential cell population called O2A cells, which are capable of forming both oligodendrocytes and type 2 astrocytes. Stem cells are a potential treatment for a wide variety of pathologies in the central nervous system. Some spontaneous neuroregeneration does occur after CNS injury, but it's often insufficient to result in functional recovery. One approach to the treatment of CNS injury is to augment the endogenous mechanisms of repair through modulation of neurogenesis. Aspects of neurogenesis that could theoretically be modified to result in functional recovery include stem cell survival, stem cell proliferation, migration of stem cells to the injury site, and their differentiation into the cell types required for repair. A variety of candidate molecules are being studied for this, with many single cytokines capable of targeting multiple components of functional neurogenesis. Potential therapies have to take into account patient age, as many cytokines important for neural stem cell function decline with age. They must also take into account the tumorigenesis, which can occur when augmenting stem cell proliferation. Another strategy is to transplant stem cells into the pathological area of the CNS to replace cells that are lost or dysfunctional. A variety of stem cells have been studied as candidates for transplantation in the CNS, including embryonic stem cells, neural stem cells, induced pluripotent stem cells, mesenchymal stem cells, and also olfactory and sheathing cells. Embryonic stem cells are limited in use due to their pluripotency and resultant neoplastic capabilities. They're also limited by ethical constraints associated with their harvest and use. Although a clinical trial for transplantation of embryonic stem cell-derived oligodendrocyte precursor cells is being conducted by Asterias Biotherapeutics for spinal cord injury. Mesenchymal stem cells are readily available and not limited by ethical constraints, but they are limited by their relatively weak differentiation potential. Induced pluripotent stem cells are an attractive candidate for CNS transplantation due to their lack of ethical constraints. Like embryonic stem cells, they require differentiation prior to transplantation to avoid tumor formation. Also, the requirement for exogenous gene transfer in the creation of induced pluripotent stem cells raises questions about the safety of their use in clinical applications. Neural stem cells tend to differentiate into glial cells, and astrocytes in particular, after transplantation into the central nervous system. This despite their ability to generate neurons in vitro and neurogenic regions in vivo. Modulation of the site into which the cells are transplanted may be important in determining this fate, and some have created artificial niches in order to influence neural stem cell differentiation after transplantation. Stem cells can also be engineered to deliver therapeutic compounds to a targeted area, as stem cells have an inherent tropism for injured or diseased regions of the CNS. In 2006, a group led by Shinya Yamanaka won a Nobel Prize for their work reprogramming adult senescent skin cells to become pluripotent by inducing the expression of 4 genes that function as regulatory nodes of the embryonic stem cell fate. In similar fashion, one can imagine that a set of master genes can be engineered into cells of the central nervous system to direct cell fate in vivo and drive the formation of therapeutic phenotypes in the absence of cell transplantation or culture. A number of studies have demonstrated that several neuroregulatory genes can convert cells in vivo to directly develop into neurons. Depending on the genes delivered, the targeted cell can be a neural stem cell, glial progenitors, or astrocytes. Lentiviral delivery of the transcription factor NeuroD1, for example, was shown to convert reactive astrocytes into functional glutamatergic neurons in the adult mouse cortex. SOX2 has been used to transform astrocytes into neuroblasts, which later differentiate into neurons in the brain and spinal cord. Striatal astrocytes, NG2 glia, and oligodendrocytes have been successfully reprogrammed into functional GABAergic striatal neurons. Mueller glia in the adult mouse retina have also been reprogrammed to a variety of retinal neuronal fates in vivo. Although many barriers remain, this approach brings us to a new frontier and points the way towards regeneration of neurons and circuits to repair the central nervous system. In conclusion, neurogenesis has been shown to take place in the adult mammalian brain, particularly in a handful of neurogenic niches, such as the subventricular and subgranular zones. Despite this, neuroregeneration in the adult central nervous system is often inadequate to result in functional recovery after injury or in response to other pathologies. Further work is needed in this regard before stem cells can be harnessed to treat central nervous system disease. Strategies for future work include transplanting stem cells into areas of pathology to replace cells lost or damaged, fostering endogenous mechanisms of stem cell repair, and also using in vivo engineering of stem cells to treat disease. And here are the author affiliations.